Good afternoon. This is Dr. Dan Guerra coming to you from Authentic Biochemistry Studios in the Inland Pacific Northwest of the United States. Today is the 3rd of October, and we're going to be following along on part 2 of my immunodiav ontological framing of the aging human. Now, we have gone quite a distance in describing to you the immunological ramifications of aging. We call that senescence at the cellular level. And we've talked a great deal about immune responses that seem to be implicated in that mechanism of aging, both at the cellular level and also at the entire organism level. And last time in the video, the first video of this series, because several of those lectures had been, of course, on the audio podcast on that feed. In the last lecture, we did the video, and I reintroduced my event ontological perspective on how to look at research that has been published so that you can formulate a trigonal planar that is coming at it from 3 different directions and trying to focus on what the scientific research tells us that will imply a potential to generate a deduction that will lead to an hypothesis that can, of course, be utilized to formulate experimental design. So the experiments are conducted both with positive and negative controls. And then after those experiments have been conducted with a high level of precision and accuracy, you generate data. And the data then will be further analyzed by the researcher, usually more than one researcher, like say a postdoc and postdoc advisor, technicians, of course, and graduate students. And then after careful analysis of the data, determine whether or not any of it rises to the level of evidence to either support or not support the original hypothesis. After that, the real work comes in, the contemplative work, where you take that evidence and try to explain it within the framework of what has already been published and what may or may not have resulted from the new experiments that you just conducted that ended up published in that paper. And that's not even enough to really understand what's going on in research science in a specific area. Let's say how the immune system does or does not have a major role in the aging process, right? Because beyond that, you have to not just look at one paper, you have to be able to study the scientific literature, which is a tremendous task. There are thousands and thousands of papers published on this particular topic, for example. So you can spend some time doing a search in PubMed and using certain search terms and look at certain types of journals that you think will have the highest quality of research and a certain maybe temporal aspect to it, so that you look at the work that goes far enough back that you look at the original studies. Those tend to be very good, very clear-minded. Because once you get into anything that is after the original research, it's going to be copying not just some of the experimental design, it's also going to be agreeing with those original hypothetical deductions often, unless it's a paper that's determined to refute previous paradigms. And that happens too, of course. At any rate, you are walking into any research area with an already largesse of information, some of which has been considered relatively pure enough to be called knowledge on a subject, right? Knowledge being justification of true beliefs, right? That's the standard definition I use. So I told you about how I do the dialectical event ontology last time. I'm not going to explain it again today, but I want you to know that that's where we're headed, like I did with autoimmune diseases, and I gave you an example of it last time. I really recommend you listen to the first video podcast, which was just a couple days ago, so that you can follow this one with this one and see how the two are hopefully continuing the framework of the discussion with precision. So let's get started now with this particular today's lecture. All right. So again, I'm calling this immunodiavantinological framing of the aging human. This is Roman numeral 2. That is the second video. I have already published, I think now about 14 audio. Those are usually about 27 to 29 minute segments on authentic biochemistry, the podcast, talking about aging, talking about the immune system in aging. Those also should be listened to so that you can know what we've already gathered from the scientific literature. We've probably covered 100 papers in those lectures, maybe more. So I'm going to continue on with that. Remember that what I do here in authentic biochemistry is I look at the published peer-reviewed scientific literature. I don't look at magazines. I don't look at newspapers. I do use review articles because they help me use a synoptic approach to be able to look at an entire, let's say, corpus of information. And usually some review articles are better than others, but usually review articles do help you to coalesce the information. And then from that, you can look at individual original research papers that are coming out of that particular window of understanding in a given field, for example, aging. Okay, that's if you've been following along with Authentic Biochemistry, the podcast, the Vera Med lectures that I've been doing now for 2 years, you know that that's what we do here. We look at published peer-reviewed scientific literature and only that. And then I try to look at the information, synthesize it, integrate it into a lecture, and then provide that lecture where we do a discussion in real time over the internet. Okay. So that's me, Dr. Dan Guerra, and that is my email address. I'm willing to take on any email that is a question about what we do here at Authentic Biochemistry and answer your questions next time if that's the appropriate format to do it in. All right. So last time we finished talking about this Journal of Neurochemistry paper published very late in 2019. So this is about 9 months old. And you can see the page numbers there. There's your citation. So let's go through this quickly because I left off with it last time. The paper wants to tell us the following. Human aging results in gradual loss of normal functioning. Okay. Changes at the cellular level are believed to contribute to that process. See, these are coherent generation of arguments to get the reader who's reading this paper to understand where the authors, the scientists, are coming from as they designed whatever's going to follow in this paper. Okay, so aging within the human brain is of considerable interest. Yeah, duh, right? Because age-related changes are frequently irreversible and can lead to changes that severely impair normal activity, all activity, including intellectual activity and physical activity, of course. Furthermore, the changing cellular environment of the aging brain is the single largest contributing factor to the increased likelihood of developing, for example, a neurodegenerative disease, such as Alzheimer's disease. Now, that's in contrast to something like glioblastoma, which is a specific type of oncogenic event, a cancer in the central nervous system. And we've talked a great deal about glioblastoma over the last couple of years in my lecture series. And you go back and look at those lectures. And we will probably impact on that again, because remember what I'm doing for aging. I'm talking about the difference between senescence and proliferation. It's going to come up real soon here, why those are important. If you have a constant proliferation of cells that are just self-divisions, and you have those cells that are of an aging population, you're going to be picking up more mutations. Mutations can arise just spontaneously over time because of reactive oxygen species being generated, because of the diatomic oxygen that's in the atmosphere, because we're aerobic organisms. And so DNA can be damaged, and DNA then can pick up mutations, right? So if you continue to replicate those cells, beyond which time they should start to senesce, you can start proliferating mutations. And if it happens in the right locus, right gene locus, it can actually result in proliferation leading to tumorigenesis. Right? And there's a lot of other factors there, but that's a general understanding of why that these two opposing forces, right, senescing, slowing down, aging to the point finally of death, or continued healthy cell division, even when the cells themselves have picked up mutations, which should no longer be replicated because they can indeed lead to then autonomous, unsustained cellular growth leading to a tumor. Okay. All right. But Alzheimer's disease is something different. That's neurodegeneration. And we've talked a lot about that too. I spent a lot of time on AD, pathophysiology, pathobiochemistry, and other lectures, which you can look up on the internet if you like. So the pathobiochemical cause of aging is limited. Okay. So what do I mean by that? Our population currently experiences unprecedented long life expectancy. Now, when I say experiences, that means what I'm trying to say is that there are expectations. That's why I call it expectancy as well. Not only do we look at what the average lifespan of a man or a woman is in 2020, people then expect that they're going to reach that average age or maybe go beyond it. Unless, of course, they get in a car accident or some other kind of non-pathobiochemical event, taking them out earlier in their life, right? But I'm saying that this life expectancy that people are picking up on is unprecedented in that people now look to medicine and then the research that supports medicine. Remember, medicine itself is not a science. Medicine is a practice. The science behind it is physiology and genetics and biochemistry, to name a few major disciplines. But medical doctors themselves, unless they are practicing scientists, are not scientists themselves, right? So science often is way ahead of what's going on in medical practice. And now that includes, of course, what's going on in pharmaceutical research. Ultimately, that leads to the clinic, maybe sometimes many years later, or it doesn't. Not so much because the research that's being conducted isn't useful, perhaps, for the practice of medicine, but because it's not noticed or it's not earmarked to be followed sufficiently to end up making it to the clinic. Right? So that means that often the medical profession is unaware of what we scientists spend all of our time doing, which is research. And they're focused on primarily what seems to be gathering evidence that has been reasoned to support some kind of problem in health. Right? So that's just a long-winded way of saying that the expectancy of life and the way that people look at it in terms of aging is a very narrow window of expectation that may or may not be supported by the primary scientific literature. And that's what we're doing here. That's why I'm following along this arc of discussion on aging and the immune response. Okay. All right. So, therefore, there is a great need to more fully understand the aging processes in order to circumvent those aspects which initiate neurodegenerative diseases. People are very concerned and worried, and naturally so, about Alzheimer's disease and Parkinson's disease, to name a few. Because if you live long enough, you've led a full life, those two diseases can be frequented as you age. However, cardiovascular disease and cancer are much more likely to kill you than Alzheimer's disease or Parkinson's disease. Most of the problem with those latter diseases, neurodegenerative ones, are way late in life, and they involve a lot of terrible morbidity in the lifespan that does continue on, right? However, it's very important to study neurodegeneration because it can happen throughout life. In fact, indeed, it does. But maybe there's repair processes that make those degenerative processes less likely to be noticed until later in life. This is one of the research areas that we've looked into in these lectures. All right. So we want to look at cellular aging, and this paper tells you that they think that if you look at cellular aging or senescence, it should give you a window to understand the organismal aging process of actually getting older as a human being, and what kind of functions at the biochemical level and physiological level and genetic level are occurring that results in the aging process, so that the person who is 65 now is not the same person who was 35 30 years ago. And so you change over time, but even within a population, or even looking, let's say, at people within one family, not all people age at the same rate, nor do they pick up the same diseases necessarily. Although there are genetic predeterminants, they're not always followed. Right? So biological research science is not something that is strictly tautological. That is, it is whatever you're able to find out is trivially true at the end. Not the case. Biological systems are far more complex. There's a lot of variation around every mean in research science and biology. Of course, biology is a general parent company for medicine. Right? All right. That's really important to understand. Studying cellular senescence is a really good tool. That's what we're doing here. A lot of the literature talks about. But you need to look at the whole organism. In particular, you have to look at the human aging process, not necessarily just looking at rodent models and trying to make that extrapolation, although rodent models are very useful. So for the brain, all of this I just said implies an understanding of how the molecular processes in neurons and other cells change as we age. So those other cells in the brain are glial cells. So primarily you have neurons and glia. Okay. There are other cell types too, but those are the two major cells in the central nervous system. So as we age, and how those interactions with those cells are affected by, sorry, I got to move this box, the changes that occur at the cellular and tissue level, as impacted by the environment, generating neuroimmunoepigenetic changes in gene expression, which can ultimately relate to the morbidities and then the ultimate mortality of the entire organism, the human being. So, glial and neuronal senescence can be understood by proteostasis. So, what do I mean by proteostasis? Obviously, stasis has to do with a centered consistency in the level of protein that occurs in any given cell lineage at a timestamp heuristic, right? So these are all events. Everything in living system are event ontologies, not substance ontologies. But we want to know, for example, what the proteome is of a given cell lineage. That is, what are all the proteins that are expressed in a given cell lineage at any given time that we're studying something, a given disease state, let's say, like Alzheimer's disease. But knowing that that changes over time. Yet, proteostasis, even though it sounds like a fixed system, as long as you look at proteostatic changes over time, you get the event dynamic ontology. That's what you need to study real biology. And this is all found in the scientific literature. You just have to make sure you read a sufficient amount of the literature to obtain even a very small window in understanding the largesse of knowledge that we have on a given topic, something as global as aging. So, proteostasis. So what I mean by that is how polypeptides, proteins, are first of all transcribed from RNA, and how the RNA is transcribed from DNA. I mean, translated from RNA. So the protein is translated from the RNA to polypeptide. That involves ribosomal machinery, either cytoplasmic, polyribosomes, or those ribosomes you find, for example, in the endoplasmic reticulum, or where genes are actually transcribed and translated directly from the mitochondrial genome or chloroplast genome in those subsystems. So you've got transcription, that is RNA from DNA. You've got translation, that is polypeptide from messenger RNA, and transfer RNA, ribosomal RNA, and a host of proteins. You've got post-translational modifications, such as glycosylation, acylation, prenylation, and then the more static responses and dynamic responses, static and dynamic working together. By that I mean both plastic and elastic elaborations of post-translational modifications. Those include things like phosphorylation and acetylation and methylation, to just name a few, that can occur to proteins after they have been even post-translationally modified and even translocated to specific locations in the cell. So by translocation, I mean the protein is made in one subcellular compartment, let's say the cytoplasm where polyribosomes are functioning, where a bulk of the proteins are translated, of course. But then those proteins, once they're translated, are modified so that they end up in organelles, like the mitochondria, or the peroxisome, or even in the Golgi. Usually, if they're found in the Golgi, they started out being post-translationally modified in the ER, endoplasmic reticulum. But some proteins, lots of proteins, also make it into the nucleus. And then there's other proteins that completely cargoed out of the cell. And some of them stop in various membranes, so they're endomembranous proteins. We talk a lot about those as lipid biochemists. That's my specialty, lipid biochemistry. But proteins sometimes leave the cell, such as pro-inflammatory cytokines, such as chemokines. So they're secreted, such as immunoglobulins. Those are secreted proteins, right? All that translocation process is something else that needs to be incorporated into the larger framework of proteostasis. And then, of course, the proteins are not immortal, they're degraded. That is, they're broken down usually by some kind of process that tears down a protein both endoproteolytically and exoproteolytically, sometimes with the help of a proteasome and an ubiquitin pathway, ultimately leading back to just amino acids, which can be, of course, sopped back up by transfer RNA and then used to retranslate a polypeptide from an RNA, a messenger RNA. Right? And also those amino acids can be used for bioenergetics, right? Or the production of other interesting organic molecules, for example, the pathways leading to hormones, the catecholamines, for example, come to mind. Okay, so similar phenomena be described for lipids, that whole process. Now, lipids are not carried through the procedure of transcription, translation, etc. But lipids are synthesized by enzymes that have gone through that processing. And lipids themselves are dynamic. They turn over and they're remodified and retailored into membranous systems or sometimes as free active lipid components in the cytoplasm, or translocating or targeting throughout the cell, or indeed outside the cell, such as things like platelet activating factor or oxygenated fatty acids like the eicosanoids. They leave the cell too, right? All right. So lipids are another really important issue, and we talk a lot about lipids because I'm a lipid biochemist. But right now, I just want you to remember that there are at least two systems that follow that. Okay, all the proteins have to carry through an intensive genetically programmed process, and all the lipids have to go through a genetically programmed process. But that genetic programming is itself both a plastic and elastic event. In other words, it can change and it can be altered by the environment to where that alteration is semi-permeant, maybe permanent, perhaps for the entire lifespan of that cell, or it can be quite elastic and the changes can come off and on. And even as a researcher, you may not notice them unless you have real-time kinetic studies to determine whether or not something has changed, such as a methylation pattern. Cytosine residues and CpG islands as promoter regions of genes that are being expressed during chromatin remodeling. Same thing with acetylation of histones during chromatin remodeling, or ubiquitinylation. So you get the idea. All right. So the senescence of the central nervous system glia is likely associated with neurodegeneration. SASP, remember that's the senescence-associated secretory phenotype. Okay. And concomitant pro-inflammatory sphingolipids and cytokines, while controlling for pathogens or tumors, may become autoimmunogenic. Right? So right now we're getting into quite the detail, right? All of a sudden, right? We're saying that there are cells that generate a phenotype, this SAS phenotype. They become secretory as they're senescing, these cells, let's say in the central nervous system. That means they can signal broad distances, right? Because they're secreting substances. So even the cells that are adjacent to them may or may not be responding to what's secreted, because you're generating, right, a gradient of information from the source, the SASP cell, to some other location, for example, the central nervous system. So even a simple first look at neurodegeneration, you have to keep in mind that glial cells themselves can become senescent, and they can generate very important bioactive proteins such as cytokines, pro-inflammatory, or maybe even anti-inflammatory cytokines. And of course, a whole host of lipids, which are signaling, which can also relate to inflammatory responses, all from cells that are sitting in situ in the central nervous system that have aged, such as the glial, microglia in particular. But the neurons do the same thing. The neurons can also become SASPs, right? They can become senescing secretory phenotypic cells and thus generate their own signaling compound, right? And that compound then can act as a base of operations, yielding changes in other cell lineages farther than just from the proximal near associated cells, but wherever the circulation will carry whatever's been secreted, such as a pro-inflammatory cytokine. So it's much more complicated. That's why I say dynamics is important. That's why you have to think like a Whiteheadian event ontologist when you think about biology. You can't think about substance ontology, like this compound, that compound, this protein, this sphingolipid, this glycerolipid, this proteasome. You have to think about all of the discussion we have in biochemistry and physiology and genetics and epigenetics as a dynamic system that changes over time. Right? That's the basis. It's actually the foundation of life. And that's in fact what aging is tracking. Okay. So you get all these pro-inflammatory compounds that are being secreted by these SAPs. Now, case in point, in a murine tau-based model, now tau is a protein that's involved, of course, in AD neurodegeneration, right? You have the A beta protein, the amyloid plaques that you've heard about in Alzheimer's disease, but you also have the tau protein, right? And the tau protein makes those fibrillary tangles. You see both fibrillary tangles and amyloid plaques associated, at least in the early studies of Alzheimer's disease, starting going way back into the late 80s, as being composite, recognizable phenotypes within the central nervous system associated with AD. Right? So the case in point, in a murine, that means a mouse model, tau-based system for AD neurodegeneration, the cell cycle arrest enzyme, which we've talked a great deal about already, P16-INK4A, it's a protein. If you take that protein, if you look at the gene that codes for that protein and use the promoter for it, so remember this is how this is done. You isolate a gene and you do a sequence analysis on it. And the gene is made up of exons and introns in eukaryotic systems. And the exons actually become coding regions that ultimately get transcribed and then translated after splicing and modifications of the RNA, such that you make a polypeptide, right? And that polypeptide has multiple functional and structural domains and all that. Well, the promoter region of that gene, like for example, this P16-4A, can be used in transgenic research. You isolate where the promoter is because the promoter has certain molecular signatures, certain regions of the DNA that tell a researcher, this is the promoter for this gene. A promoter, I don't want to belabor this too long because I assume most of you know what all this means, but a promoter is that region. Often it's 5' to the open reading frames that will end up being the exons that end up being spliced together to make the polypeptide after transcription and translation. But the promoter is where you get RNA polymerase and a whole host of other proteins to bind to the DNA, unwind the DNA by acting on topoisomerases and gyrases and having that RNA polymerase 2 in there, single-stranded binding proteins, ultimately being able to make a messenger RNA, right? All of that processing has to occur during chromatin remodeling for the transcriptional process. Well, that promoter is something specific to any given gene, although it has regions of homology to all genes, because those proteins I just told you about that are involved in transcription, of course, work on all of the promoters on all the genes that get expressed in any given cell lineage. Okay, so they took that promoter and they drive. So they used the P16 Inc. 4A, promoter-driven destruction of senescent glia. And when they did that, they blocked tau pathology. So what does that mean? We already know that this protein's involved in senescence, right? So if you use the promoter of the gene and you drive the expression of two different kinds of genes, one is a reporter gene, so you can study its expression, and the other is, you could call it basically a suicidal gene, that once it's expressed, it will kill those cells. You can ask the question, first of all, where are these genes that are normally expressed from that promoter found, right, such as the SASP cells? And once we eliminate those cells from that lineage, such as the senescent glial lineage, what does it do to tau pathology? In other words, are they linked? Are they associated? And in this particular study, it was determined that they were linked. So getting rid of senescent glia blocked the normal pathology of the tau protein, which ends up being those fibrillary tangles, which is one of the phenotypes of the cellular degradation process, neurodegenerative process that ultimately yields Alzheimer's disease. Okay. So, developmentally, neurons in macroglia, like astrocytes and oligodendrocytes, share a similar embryonic origin deriving from the neural tube, of course, and they all arise from the ectoderm. This is in terms of early development of where these cell lineages come from. Now, on the other hand, macrophage-derived microglia, which you find in the central nervous system in huge abundance, enter the brain following directed migration and take up residence before the blood-brain barrier has been formed. This is in utero, right, during gestation, right, of the developing fetus, of the human. Now, it's unclear if microglia are bone marrow origin, like a normal macrophage, but it is currently established that they have a very primary origin, likely derived from the yolk sac macrophage precursors, and as distinct from naive blood poly or mononuclear cells. So they remain quiescent, yet surveilling. They do the surveilling. They look for foreign antigen, for example, or foreign organisms, right, like macrophages. They do surveilling until finally activated as a threat to adjacent neurons is detected. So that's how these microglia work. So you have to know something about their origin to understand how alterations in gene expression, including ultimately becoming senescent, is going to follow a totally different trajectory than neurons, even though they're sitting in the same system, central nervous system. Okay, so perivascular, meningeal, and choroid plexus macrophages also reside at the CNS interface, and the peripheral macrophages can penetrate, indeed still, once it's made the BBB, the blood-brain barrier. So there's more possibility for macrophage accumulation. Now, having said that, microglia are not formed from myeloid progenitor blood cells. Okay? Rather, they increase through cell division, through mitosis. Okay? So that means that their programming for cell cycle is going to be on a different temporality and is going to respond to different signaling phenomena than the neurons are. Right? And they should be, because they have a different function. See? And they have a different origin as well. So since the microglia are predefined early in development, they age accordingly, and thus they may become SASP resident loci with the potential to associate with neuropsychiatric and neurodegenerative diseases without any necessary collinearity with the neuronal temporal changes in gene expression, which are also going to be august and profound, right? Because of the whole process of neurogenesis and the synthesis of synapses, dendritic fibers, and whatnot. They're going to allow the axons and the neuron and the synapses to be formed and reformed over time. And all that process is not going to be directly the same lineage as going on with the microglia, although microglia are involved in those processes, doing their other part-time work, right? Microglia also aid in the process, for example, of Bouton formation and synaptogenesis, as do other glial cells. All right. Now, microglia in the aging brain are senescent microglia the key to neurodegeneration? It's a question that's asked in this paper here. This is from the Journal of Neurochemistry, the same journal we'll be looking at. Just make sure that you see it down there because it's in light blue. So, microglia in the brain, of course, promote repair of what? Of neurons. They're involved in the synaptic pruning. That's what I was going over, rather roughshod a moment ago. Synaptic regulation, how all of the various neurotransmitters are organized and associate in metabolomic or chemotropic responses at that synaptic junction, right? They recruit other cells, so microglia can recruit other cells that are associated with a damage response, or perhaps even with a repair response, or even in co-organizing networks of neurons to fire together. Right? They phagocytose because they are like resident macrophages. So they phagocytose debris and foreign material because you can't leave that material around if a cell dies. So this is their normal housekeeping job. They will release proteases and reactive oxygen like any macrophage does. Proteases are going to be degrading any polypeptides that are not intracellular but extracellular, and sometimes those proteases are altering the extracellular matrix, right? That glycocalyx, the proteins that are glycosylated that make up the matrix that are associated with all the cells within a given tissue system. And those proteases are functioning to alter that matrix, that extracellular matrix, to affect new processes like synaptogenesis, which must require some proteolysis, right? That's what those proteases are for. Reactive oxygen also it can destroy, because of the introduction of partially reduced oxygen into DNA and RNA and protein that may be errantly in the extracellular matrix, therefore suggesting to the system that there are foreign invading pathogens or relics or remnants of those foreign or invading pathogens, or, for example, virus. So they eliminate damaged neurons. Neurons age, they die, they can be damaged by stress, they can be damaged by alcohol intoxication, by pharmaceutical drugs, by illegal drugs. Various processes can damage neurons. And those neurons, once they're damaged, need to be taken out. That's a major function of the microglia. They also release cytokines most of which normally are attributed to being pro-inflammatory, but there are also a host of anti-inflammatory cytokines that can be generated to suppress an immune response. Okay. They also are involved in neurotrophism. That is how different neurons aggregate together and fire together. We've already mentioned that. And of course, the general process, just like any macrophage, is survey to carry out surveillance of the entire cellular environment via signaling. So, for example, post-trauma infection, neuronal dysfunction, or CNS fluidity changes, including pressure alterations, membrane topodynamics, as well as drugs and xenobiotics, the microglia migrate to that site of injury and convert to an activated status. That's how they are turned back on, right? So non-phagocytic reactive microglia, so at this stage you're not being phagocytic, display a thickening of their branches, an upregulation of the MHC class 1-2 expression, secretion of the pro-inflammatory cytokines, and more reactive oxygen is being made. And they can assume a phagocytic state subsequent to that activated state, and coincidental with it, and obtain a phenotype, and with the phenotype is a professional antigen presenting cell, to phagocytize the material and display it for what? Resident T cells, like the innate-like T cell lineages, type 2 that we talked about back in Authentic Biochemistry podcast lectures previously. So there is also evidence that microglia can support remyelination following axonal damage. So you have myelination of certain axons. We talked about that at great length. It enhances the saltation process for rapid transmission, neurotransmission, right? Remember that the myelin itself is composed of sphingomyelin, which is an important sphingolipid, and it is implicated in neurodegeneration as well, and also and in repair, as in this process we're mentioning here. Look where the microglia are involved in carrying out associated remyelination of the axons, of the neurons associated with networks that are involved in neurotransmission. In fact, depletion of microglia in experimental models of remyelination showed significant impairment, implying that there is a very important role for microglia in remyelination. Microglia may polarize into the pro-inflammatory M1, like M1 macrophages, or the reparative phagocytic type, just purely phagocytic M2, which tend to actually produce and generate and secrete anti-inflammatory cytokines, much like a T regulatory cell might do in the T cell population. So the phenomenon is not that simple. It's more complicated than the macrophage distribution. Human CNS microglial transcriptional profiles that have been looked at, what genes are expressed at the RNA level first, reflect a constellation of transcription factors first synthesized, chromatin remodeling sequelae subsequent to all of that, and that may actually be a family of discrete cell lineages, perhaps more like T lymphocytes, okay? So once you have this remodeling sequelae and that gives you all these diverse functions, it might be that the microglia themselves are a family of discrete cell lineages, depending on the networking and communication they get with the rest of the central nervous system. Right? So they are unique upon themselves. And then those cells, based on the activation or suppression, can proliferate via division, thus changing the entire central nervous system in situ, while aiding in the process of neurotransmission, pruning, and all the other functions I just told you. So they're very dynamic systems. Right? So again, it may suggest microglia may actually be a family of discrete cell lineages, perhaps more like T lymphocytes than innate monocyte macrophage terminal differentiation motifs. That's a good argument because of all the functions we found out, the level of gene expression, transcription factors, regulating gene expression, etc. So dynamic systems. I have suggested that microglia play a role in learning, being necessary for synaptic remodeling and pruning, of dendrites, depending upon stimuli, time, and coordinate positioning, all involved in the geometry and positioning of the microglia, the relative stage of activation, and the expression of those genes. We've just been talking about transcription factors first, and then whatever transcription factors turn on from the genome of those microglia. Now remember, these microglia are also aging. So imagine how this complex, diverse, beautifully orchestrated system functioning well throughout life then starts taking hits over time because of excessive amount of reactive oxygen, damage to the system because of xenobiotics like alcohol and drugs, and then the overall senescent process, which just simply has to do with how many cell divisions any given cell can make before they stop dividing. For example, because of telomere attrition. Right? So these, in other words, the microglia start losing it over time and loosening their appropriate plastic and elastic functioning. And they themselves then can cause damage. So research literature has demonstrated that aged mice induced with a pro-inflammatory interleukin-1 beta or interleukin-12, differing priming gene prototypes, including classic, which after subsequent exposure to interferon gamma and become neurotoxic, and alternative when subsequent co-exposure to interleukin-4 and interleukin-1 beta become neuroprotective. So did you get all that? So you can have a pro-inflammatory system, right, in mice based on microglial prototypes that can be at first neurotoxic, but then subsequent to exposure to those same pro-inflammatory cytokines, this is what we saw, for example, Tregs when they're exposed to interleukin-2, remember? They can now become neuroprotective at the same time. So they deal with damage, they deal with, say, inflammation, they deal with an invading organism, they deal maybe with a tumor, but at the same time, they repeal that activity and become neuroprotective, temporally signatured. So toll-like receptors 2, 3, and 4 appear to be essential for this classical priming process, right? How the microglia are controlled. So take a look at this whole process. The transcriptome of these cells might be dependent on the high mobility group box1 gene product since its expression, inhibiting expression, blocks priming together. So you have a stress-induced hypertension. Any given stress can be involved. For example, anoxia, right? Or a small brain injury, a traumatic brain injury, something like that. The stressor can then generate this HMGB1 gene product, right? It's the high mobility group box 1 gene product, which is then going to be positively working through RAGE. Now, RAGE is another entire component that I've talked about before, so I know what it is, and I want to remind you, or tell you for the first time. RAGE are advanced glycation end product receptors. Okay, so these advanced glycation end products, right? Those are carbohydrates that have been modified via usually oxygen, sometimes methyl groups, that are associated with aging over time. So here's where carbohydrates involved. And of course, those carbohydrates have a receptor that they bind to. And this HMGB1, right, induces this receptor to be expressed and induces its activity at the plasma membrane. So there's an axis of HMGB1 RAGE. That's what's shown here, all positively induced after a stress response, say like a TBI or a drug or an alcohol intoxication. So that's going to now generate something inside the cell. So you have an impaired stress-induced mitophagy. Now, mitophagy is when mitochondria turn over. They digest themselves. That's what phagy means. So they have an impaired stress-induced mitophagy flux in these microglia. The obtained microglia-mediated neuroinflammation then is pursuant. Okay, so that's the entire process that we're talking about here. Right? So you've got stress-induced induction of HMGB1 working through the RAGE receptor, and then altering the entire mitophagosomal processing by combining with a lysosome, taking the mitochondria that's normally going through mitophagy, and then blocking that response so you don't make an autolysosome. Rather than that, what you get is an impairment of the mitophagosome lysosomal fusion. It doesn't occur because of this induction with HMGB1 through the RAGE receptor, all induced by the stress response. So what happens is RAB7, a particular gene involved in DNA repair, and lysosomal acidification do not occur. Okay, this will change now, this microglia, to turn on an M1 polarization. Remember, that's pro-inflammatory. So it's one of the transcription factors that have kappa B, right? That will then generate interleukin 1 beta. These are really potent pro-inflammatory cytokines. Tumor necrosis factor alpha. That will generate in the brain that's been induced by the stress and going through the HMGB1 RAGE, alteration of mitophagy, yielding the M1 pro-inflammatory phenotype for the microglia, then generating and secreting the pro-inflammatory cytokines, generating a neuroinflammatory sympathetic overactivation in the central nervous system. Okay. This is all occurring in the rostroventrolateral medulla. Right? This is where it's occurring in the RVLM. Now, this comes from a paper published in the Journal of Neuroinflammation very recently in 2020. So I want you to see now how you can take a resident microglia, and you can convert it into a pro-inflammatory system. So that's distinct from microglial senescence. So let's take a look at this. Now, that's an activated microglia. Now we're talking about senescence. So how do you get microglial senescence? This is another very important pathobiochemical process, which we now have to look at. You get an increase in ferritin-associated ion storage. And you know, wherever you have iron, you can generate reactive oxygen, right? Because of the valency of iron from the ferrous to the ferric, right? So you can generate those reactive oxygen, and because of that, because of all the excess iron, that's going to yield to more mutations. Okay, so you have cytorexis occurring, you have process deramification occurring. That's where you get this strange morphology change. The microglia start to swell. They can't move as much because they're senescing, so motility drops. They can't phagocytose anymore because this whole morphology means these are microglia that are going out of commission. Right? They're losing all the flavor of their normal phenotypic characteristics to be professional microglia, right? And some of the other processes occur is that ROS increases, the KV1 increases, that this is potassium channel translocating system in the microglia. SIRT1 drops, now that's a deacetylase, okay? And ultimately what you get because of these processes, reactive oxygen, potassium channels, SIRT1 diminution, right? Potassium channel kicking up here, which is inducing the swelling, by the way, because bringing water in, right? All that then causes this phenotypic change and generation of pro-inflammatory cytokines. So there's an increase in iron storage and ferritin, senescing microglia. There's an increased release of neurotoxins. There's a reduced ability to phagocytose protein aggregates, leaving the neurons vulnerable, such as with amyloid plaques and tau fibrillary tangles, you see. Altered surveillance phenotype with less dendritic branching is going to occur, and there's going to be reduced process motility, as we said. Post-stress, microglia exhibit lower migration rates, and they have a more sustained inflammatory response and reaction to damage. Right? So this is a different aspect entirely. We looked at activation of microglia and what they're doing, right? Now we're looking at senescence of microglia, and we can see that they're also pro-inflammatory. You get the idea that there's a total change here. Again, this is from that same paper, Journal of Neurochemistry. So, what can we summarize here a little bit? Low-grade inflammation, okay, occurs everywhere in the body. It's paradigmatic, though, in the aging brain. As all the organs and all the cells and all the tissue beds age, low-grade inflammation in all those systems becomes more prominent. This is because those cells are starting to generate the SAS phenotype, right? That senescence-associated secretory phenotype, which is going to yield compounds which are going to induce a low-grade inflammatory response because of the reactive oxygen increase, because of the production of oxygenated fatty acids, like some of the pro-inflammatory prostaglandins, like, for example, the synthesis of thromboxanes, or going through the lipoxygenase pathway, such the production of leukotrienes, right? This is going to generate an in situ inflammatory response. It can ultimately yield an activation of cells generating pro-inflammatory secreted glycoproteins like the cytokines. Okay, so low-grade inflammation is the paradigmatic aging brain with elevated pro-inflammatory cytokines, there you go, and the diminishment of anti-inflammatory cytokines. So you're losing that ability to suppress the immune response as the cells age, as well as amplifying the amount of inflammation. Okay, so I told you before, when you hear that as you age, you have an impaired immunity. To the layperson, they think it means that the immune response is hindered. It's not what that means. Impaired immunity means that you actually are, in many respects, hyperimmune because you have low-grade inflammation to start with. In fact, this happens with many diseases, particularly chronic diseases associated with obesity, because of all of the lipids that are in circulation associated with lipoprotein. Okay, that's another story that I have covered in other lectures. Okay, so microglia offer chronic senescence and thus neurodegeneration. That's what you get from aging microglia in the central nervous system. SASPs then, remember that's the senescence-associated secretory phenotype that can happen to any cell, not just the microglia. SASP epitomize aged mitochondria, where the SASP again is associated with pro-inflammatory cytokines and chemokines, which direct the movement of cells towards the site of inflammation, enhancing that inflammation usually if they're in such respect and the receptors are there, and so they're being recruited. More reactive oxygen, growth factors, which are going to also stimulate this process. Proteases in the extracellular matrix, which can damage surrounding cells and the matrix holding them. And it's thus, this entire SAS system, degenerative and it leads to morbidity as the central nervous system ages, and any system that has the SAS processing, and then mortality as a consequence. So you get the full picture now. Right? All right. Let me finish this last page and then we'll take a break, come back and do another lecture. So cellular senescence while closing down cell division and allowing for an opportunistic infection. Okay, that's what cellular senescence is direct result of. And the potential for an autoimmune disease because of inappropriate recognition of host factors, proteins, lipids, for example, may, when functioning properly, perform tumor suppression. So all the things that don't look good with the SASP response may actually be involved in tumor suppression because of this low-grade inflammatory response, potentially poised to determine errant autonomous cell replication where it's not supposed to occur, and then the mobilization of pro-inflammatory cytokines, chemokines, and then natural killer cells can be brought into the system, as well as T lymphocytes that can be working in the effector cell lineage, destroying whatever tissue is going through that proliferative stage. So this is an argument why, in terms of how the body has evolved, in terms of the central nervous system has evolved within the human system, and the fact that you have a brain for a long period of time, I said before, like 70, right, minimum, that's a long time to surveil and maintain the central nervous system. So in terms of the proximal events that maintain that process as one ages, having the SASP, that phenotype, may be responsible for controlling rampant autonomous cell proliferation, as in oncogenesis, tumorigenesis. That may be why it's there. At the same time, the SASP can induce damage and neurodegeneration. So remember before, way at the beginning, I said that there's this expectation that people live longer and longer because of all of us work that us research scientists do that gets tunneled into certain parts of the practice of medicine and in pharmacy and in, of course, nutrition, recommendations in nutrition, right? Ultimately, to extend that life expectancy. Keep in mind that the cells responsible for maintaining a high integrity human system, which is all organized from the central nervous system, the CNS, the brain, is already built in to become senescent. And eventually that will result in higher levels of morbidity because of the potential for infection and autoimmune disorders and chronic inflammation. But also at the same time, which we're going to get to in a minute, SASP can also promote tumorigenesis when it's incorrectly working. I think you understand that because of the way that it has this biphasic system of expressing itself in terms of signal transduction cascades to other cell types because it telegraphs long distance. So that being said, senescent cells may promote tumor progression because of a decrease in antigen-presenting cell-mediated immune destruction of tumor cells. Remember I told you that you lose that ability to become an APC? You don't have an APC, you can't target tumor cells. So old age-associated pathologies via senescence-associated secretory phenotype have already been examined by me many times in these lectures. And in my view, the basic concept is one of ratio and proportion, where, okay, this is something that's I've developed. I'm using my dialectical event ontological paradigmatic view of the research literature to come up with hypotheses via deduction that ultimately can be translated into experimental design. So this is the early stage of that process where reasoning, contemplative reasoning, taking evidence, taking concepts, and taking ideas and integrating them via contemplation can yield new levels of research that can maybe get a better handle on what aging really is. If indeed we apply it to this system, or we can apply it to cancer or cardiovascular disease or any other physiological process that may have a high level of functionality that becomes degraded over time or because of pathology and thus is required for the practice of medicine to get a better understanding of, and that's where us research scientists come in, to teach the medical doctors as well as to provide them with information and to explain it. Okay. So my view, when I've studied this literature on aging, we need to look at senescence versus necrosis. Necrosis is cellular death. Senescence is cellular aging. So I want to make a ratio out of that. That's a cell fate. And I want to set it up in a ratio and proportion, which you'll see why in a minute. So I'm going to multiply that by, once you have this overall result of senescence or death, what are the mechanisms that get you there? Well, proliferation and degeneration. So these are a component of the mechanism associated with cellular fate ontogeny. Right? All right, that's ontogeny, not ontology. Now, depending on which of the four factors is elevated, there's four factors here. may be eclipsed by mortality. Okay, so whatever's going on with this interaction of cell fate and then the mechanism to drive that cell fate, mortality can abruptly end it. Right? Or another way of looking at it, the ratio and proportion of these processes can result in the mortality. So in this way, SAS may promote or inhibit pathophysiology at the local or at the systematic level. For example, if senescence is an unknown in this process, so what we're looking at is to solve for a variable. So that's why you do cross multiplication in very basic high school algebra, to solve for a variable. Right? Now in my system, I'm trying to determine, first of all, do these variables interact with one another in situ in biological event ontologies? And if they do, am I going to be able to generate an interactome to describe how they do, right? And so what I want to do is break this down to mathematics. We've told you before that you have mathematics, and mathematics is the result of logic. So you start with logic, you have mathematics. Mathematics gives rise to physics. Physics in many ways gave rise to chemistry, and then chemistry in many ways gave rise to biology. And then us biochemists, we take and integrate what is known in chemistry and introduce it and own it within the biological systems. Right? So that's why we're biochemists. We're a hybrid scientist. And so this whole process is ultimately brought all the way down, boom, 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 to logic, right? To the logic. So, for example, senescence is unknown, we call it S. This is what I thought through, which stands for the number of SASP cells per cubic centimeter. So I'm setting up a hypothetical deduction. This is me deducing the hypothesis. And necrosis is the number of dead cells per cubic centimeter. So this is quantitative because it's mathematical. While proliferation is measured by the number of cell divisions per cubic centimeter. And degeneration is measured by the number of damaged cells per cubic centimeter. Right? So I'm taking my invented ratio proportion. I want to look at these four parameters that themselves stand in for aging, and then what occurs in aging, such as senescence and necrosis, right? A cellular fate, if you will. And then the mechanisms to get you to either senesce or to die. And these would be things like, do cells continue to proliferate or divide, so that proliferation gets to a point of oncogenesis, or are they degenerated and broken down and destroyed? So that's where I plug in the numbers. So let's say we have S is unknown. We say the N here is going to be 100. So that's the necrosis, right? The P, which is proliferation, we're going to give a numerical value of 60. Now, these are just numbers I chose to see whether or not the model seems to work. And then the degeneration is going to be 15. Right? Okay. So we cross multiply. We solve for S. Now we're solving for SASP in this system. We're solving for senescence. So the extrapolation we make is when we do this cross multiply, right, by this induction of the ratio and proportion to generate via deduction and hypothesis of what's going on in the aging brain, right? All of that, can we come up with a way to analyze cell types and determine if they do indeed vary and where is the source of the variation in these fatalistic and opportunistic alterations of the cells in the CNS. Because then that'll allow me as a researcher to design an experiment to be able to segregate and separate these phenomena and study them in more detail by keeping the others constant. That's a part of experimental design. So in this example, I cross multiply, and it gives us a number 400 SAS per cubic centimeter, okay, just by throwing in these numbers here. the numbers that actually deal with real things you can measure, right? You can measure how many cells are dead, how many cells are going through division, how many cells are, all these things are things you can measure. And the reason I chose S as the unknown here is because the senescence phenotype is more complicated. Right? It's more complicated to measure because there's such a wide constellation of senescence processing over time. And you want to catch it early enough to know that some cell lineages, for example, are starting to become senescent in certain, let's say, regions of the brain, like in the hypothalamus or the hippocampus or the amygdala, for example, right? Prefrontal cortex. All right, so this example across multiply, I got 400 SAS per cubic centimeter. That signifies the correlation of SAS to proliferation, right? So this could be used. So how we're saying that SAS seems to be associated, according to this study, if we generated those numbers and we wanted to know what our senescence involvement in this variation that ultimately leads to aging in this system, the human brain, we plug in these numbers that we've measured, but we have not measured the senescence phenotype because it's more difficult to measure. Not a single point of recognition because we don't necessarily have good biomarkers for it. With the other systems, we do much better, I could argue. This could be as an index of evidence demonstrates that SASP is associated with oncogenic event. This would require, though, a careful set of studies. So you'd have to do these experiments, right, with cells in culture, with animal models, upon autopsy of dead humans, and whatever cell types can come from neurosurgery on living humans, right? So that we can get enough data on these different cellular fates and the mechanisms that obtain us those, apprehend those cell fates. We can start getting a handle on which of these processes is actually functioning, and maybe you can get a temporality. Right? So require a careful set of studies, for example, glioblastoma, megaforming, surgical explants, or an autopsy, as I said. All right. Now, I'm going to stop here and I'm going to tell you two more like basic fundamental things. Other scenarios must be analyzed against evidence for any other disease. Here we're looking at glioblastoma. We're looking at proliferation. For neurodegeneration, we might want to solve for how many cells are going through neurodegeneration and try to be able to discover some good phenotypes. And we do have some for secretion for sure of the SASP cells, even in the early stages. Well, I showed you the morphological changes in microglia, for example, which can be measured microscopically. So, such as cardiovascular diseases, so we can completely discuss this in different regions of the body. We don't have to just be looking at the central nervous system. Metabolic disorders, right? And for pernicious infections. Right? To understand which processes are involved, where there might be an overshooting of the mark, either an autoimmune response or proliferation of cells. And so you don't get a hyperimmune response, you don't get a hypoimmune response. And you understand what could be happening to the vascular cellular bed upon induction of, say, a pharmaceutical agent. So this is where the dialectic comes in. And the dialectic is used in this expression I'm just doing here by generating a ratio and proportion and coming up with it, using that to deduce an hypothesis. Right? And then the rest of it has to, of course, be experimentation, to design the experiments. I got into a little bit there with you. But designing experiments and then generating data, finding what data can give you prima facie evidence, and then understanding the evidence in the backdrop of the corpus of the scientific literature. So that's ultimately where you want to end up. So, and that's where we're ending up right now today. All right. So hopefully that wasn't too fast or too slow. Hopefully that was a reasoned, processive lecture that gets you from the very beginning of just some basic ideas of cellular senescence and aging. And then I brought you forward enough to understand where microglia are really important. So again, why did I do that? Because I'm looking at different immune cells. We spent a lot of time on T lymphocytes. We looked at, previously at B lymphocytes. We've talked about neutrophils. We've talked about eosinophils. We've talked about circulating monocytes and macrophages, right? We've talked about all these different cell lineages. And I have way in the past, meaning like 6 to 8 months ago, talked about microglia quite a bit because I had a whole series on Alzheimer's disease. But now we're looking at microglia, and what I'm trying to give you, what I think I am establishing, is that the immune system is intimately involved in aging. Right? And so now I'm giving you the examples of how we can go about understanding this by taking the deep dive into the scientific literature. So I'm going to stop here. I don't think we went too long. This is Dr. Daniel John Guerra from Authentic Biochemistry Studios on the 3rd of October saying bye. 4